Good day, this is the Professor, Professor CBMD PhD, and today's Monday, September 12th, 2022. It's 6:09 and 25 seconds, a.m. Eastern Standard Time, and today's research, high blood pressure. So let's hop to it here. High blood pressure may accelerate bone aging. Date September 7th, 2022. Source, American Heart Association. A new study in mice found that hypertension may be linked to significant bone loss. Bone quality in young mice with high blood pressure was similar to the bones of older mice without high blood pressure, suggesting that the effects of hypertension on bone quality, which includes strength, may mimic that of aging. The researcher suggests early detection and treatment of high blood pressure may help to deter loss of bone quality as people age. High blood pressure and osteoporosis are prevalent diseases, and people may have both at the same time. In this study, researchers examined inflammation associated with high blood pressure in mice and found it may be connected to osteoporosis. The bone marrow is where both new bone and new immune cells are produced. Researchers suspect that more pro-inflammatory immune cells in the bone marrow may be leading to damage of the bone and making it weaker. By understanding how hypertension contributes to osteoporosis, we may be able to reduce the risk of osteoporosis and better protect people later in life from having fragility fractures and a lower quality of life. In the study, researchers compared young mice with induced hypertension to older mice without hypertension to assess the potential relationship of hypertension to bone aging. The human age equivalent was about 20 to 30 years old for the young mice and about 47 to 56 years old for the older mice. A group of 12 young mice, 4 months old, were given angiotensin II, a hormone that leads to high blood pressure. The young mice received 490 kilogram of angiotensin II for 6 weeks. A group of 11 older mice, 16 months old, also received 490 kilogram of angiotensin II for 6 weeks. The two control groups of 13 young mice and 9 old mice received a buffer solution that did not include angiotensin II, and these mice did not develop high blood pressure. After 6 weeks, researchers analyzed the bones of mice from all 4 groups using microcomputed tomography and advanced imaging technique. The bone health was determined by strength and density of the bone. Mathematical algorithms were used to estimate the potential effects of hypertension and aging on the microstructure and strength of the bone in the mice. When compared to the young mice without hypertension, the young mice with induced hypertension had a significant 24% reduction in bone volume fraction, an 18% reduction in the thickness of the sponge-like tubercular bone located at the end of long bones, such as femurs in the spinal column, and a 34% reduction in estimated failure force, which is the ability of bones to withstand different types of force. The failure force translates into weaker bones in the spine. Bone weakness can lead to vertebral fractures later in life. In contrast, the older mice who were given the angiotensin II infusion did not exhibit similar bone loss. During the study, however, the old mice, with or without high blood pressure, exhibited a reduced bone quality similar to that of the hypertensive young mice. In these mice, being hypertensive at a younger age essentially aged bones as if they were 15 to 25 human years older. And to assess the impact of inflammation on bone health of the mice, researchers analyzed the bone marrow using flow cytometry, and this tool allowed researchers to identify individual cells and to sort out specific immune cells. In the hypertensive young mice, they found an increase in the number of inflammatory signaling molecules, indicating an increase in inflammation in the bones when compared to the young mice that did not receive angiotensin II. And this increase in active immune cells tells us that the older mice are more inflamed overall, and that a continued state of inflammation, whether they have high blood pressure or not, may have an impact on bone health. It appeared that high blood pressure was adjusting the bone remodeling process toward bone loss rather than bone gain or bone equilibrium in the hypertensive young mice. As a result, bones will be weaker, leading to an increased risk for osteoporosis and fragility fracture. In humans, this might mean that we should screen for osteoporosis in people with high blood pressure. The researchers said that these findings may help researchers identify the immune cells and mechanisms that play a role in human bone health. And this depth of knowledge may lead to new approaches to prevent osteoporosis as early adulthood. The study limitations include that it is only descriptive, so additional research is needed to investigate how specifically the different types of immune cells may contribute to bone loss. In addition, it is unknown whether a similar link exists in humans, so similar research in humans is needed to confirm these findings. Good day, this is the Professor of Microbiology, Bioinformatics, and Pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.